So what I want to do here is to begin a discussion of the structure of transmembrane proteins. Well, you remember that what we had to do is we had to think about the idea of having a phospholipid bilayer, a relatively impervious layer of phospholipids organized in two layers of phospholipids. The question then was, how are we going to manage to communicate from the outside to the inside? That after generating this formidable layer, then we needed to have some mechanism to communicate outside to inside, and that communication, we said yesterday, revolved around the idea of having transmembrane proteins. But the first question we need to ask here is, how do we manage to put a protein across this phospholipid bilayer? And it turns out that on the inside we can have down here the amino terminus. But then when the protein approaches the membrane, it grows across the membrane as an alpha helix. So proteins traverse the membrane as an alpha helix. Well, there are a couple of points I want to bring up here. First is that recognize that the alpha helical structure is embedded in this hydrophobic region. The fatty acid side chains, which are hydrophobic, are interacting with the amino acids that are spanning the membrane. The second point I need to make here, and I misspoke yesterday, is that each time you step from one amino acid to the next, the advance is 0.15 nanometers. I said 0.45 yesterday, and that's simply not right. It should be 0.15. Now, the number isn't important. But if you imagine this is a spiral staircase, and if you imagine you're climbing the spiral staircase to go across the phospholipid bilayer, and you imagine that each step takes you 0.15 nanometers, and if you recall that the thickness of this membrane is in the neighborhood of 6 to 7 nanometers, you now see that you have at least 40 amino acids that are required to be in this spiral staircase that goes across the membrane. You'll also recognize that because these 60 or these 40 or 50 or so amino acids that are spanning the lipid bilayer are embedded in the hydrophobic region of the membrane, that these amino acids are predominantly hydrophobic. And in fact, in the early days when they were beginning to sort out the structure of these transmembrane proteins, what they would look for is regions of hydrophobic amino acids and then signify those regions of hydrophobic amino acids, usually 40 to 60, as the transmembrane regions. But these are the amino acids that go across the membrane, and I'll add this in. I'll never ask you to memorize this, but I think it's a handy sort of tool. It's called malic. And these are the single letter amino acids that describe the amino acids that are most likely found in the transmembrane region. This is methionine, alanine, leucine, glutamic acid, and lysine. Now, don't memorize that. But I just think it's kind of convenient that there's this nomenclature of Malik, which tells you that you have predominantly hydrophobic amino acids. And if you drill down into this, you find that they have to have a certain flexibility so they can form the spiral staircase. And you'll notice, though, what I've done. You'll notice that I've put the amino terminus on the cytoplasmic side of the membrane, and I've left the carboxy terminus in the extracellular space. You'll notice that that's a single span across the membrane. Well, it also emphasized yesterday that what we really need for these transmembrane proteins are proteins that generate a kind of channel, a kind of hydrophilic pathway that allow ions to move from the outside to the inside and the inside to the outside. If you look at this structure, you'll immediately recognize that with these hydrophobic amino acids spanning the lipid bilayer a single time, there is no hydrophilic pathway. There are several proteins that do this. They come across the membrane a single time. They have a single alpha helix across the membrane. But more commonly, what we see is that a protein will span the lipid bilayer several times. And where I'm going to go with this is we're going to talk about how we can take a protein, thread it across the membrane several times, and then organize those alpha helical regions and employ certain R groups on these alpha helical regions to make a hydrophilic pathway. So that's where we're headed next. Any questions to here? So now I'm going to redraw this. And we're going to first view it as we have over here, sort of as a cross-section, as though we've cut through the lipid bilayer. We're looking at each of the leaflets of the lipid bilayer. This is out, this is in. And we'll start here with the amino terminus. We'll run a region, probably some beta-pleated sheet, and then we enter the membrane. We cross the membrane, and I'm going to label this as 1. Then out here in the extracellular space, these proteins can enter beta-pleated sheet. They can be some alpha helix, beta-pleated sheet. This now just begins to generate the three-dimensional shape of this protein in the extracellular environment. So in addition to the alpha helix and the beta-pleated sheet, there will be R groups on these amino acids, some of which are hydrophilic, some of which are hydrophobic, which will determine the three-dimensional shape of this region of the protein in the extracellular space. Because after all, out here we're living in an aqueous environment. But then this protein will take a turn. It will then run another alpha helix across the membrane. There'll be a cytoplasmic region in here, another alpha helix, an extracellular region out here, and now I'm going to show this as a fourth spanning region, and down here is the carboxy terminus. Outside of this will be the phospholipid bilayer. I'm going to number these now as 1, 2, 3, and 4. I'm simply numbering them so we can keep track of those individual alpha helices. Because in this image, we've taken each of these alpha helices and spread them out so we can see how they cross the membrane. The next thing we're going to do is we're going to imagine putting our head into the board, or alternatively taking the structure and turning it face on and looking down on the surface of the membrane. What we want to think about is looking down onto the surface of the membrane and recognizing the orientation of these 4 alpha helical regions. And then what we're going to do is we're going to rearrange these alpha helical regions, which we've spread out and made flat, and look at them as though they're surrounding a central channel. So, these are spread. Now over here, I'm going to draw circles. Each of these circles is meant to represent the phosphate head group. These are the phosphate head groups. And depending on how you think of it, you can imagine putting your head into the board looking down, or you can imagine taking this cross-section and rotating it from this orientation to this orientation and looking down onto the surface of the cell. Well, when we do that, now we have to rearrange these a little bit so we can see them, and we're going to now show this as an orange circle. This is the first alpha helix. That's the second, the third, and the fourth. So now we're going to number them 1, 2, 3, and 4. So all we've done here is taken this splayed out view of the alpha helices and brought them together so that they can interact one with another. Now imagine as you look at each alpha helix, 1, 2, 3, and 4, and you're looking down, imagine this circle extending down through and into the plane of the blackboard as a spiral staircase. The spiral staircase of amino acids as we're walking down across that membrane. I think you can see as you walk the spiral staircase, some of these amino acids are going to be facing the hydrophobic fatty acid side chains. And some of them will be facing inward toward one another. So you can imagine 4 spiral staircases going across the membrane, and each of those steps having a side group that's either hydrophilic or hydrophobic. I think you can see from that, at least in general terms, that the R groups that are going to be out here facing the phospholipid bilayer, those that are facing the phospholipid bilayer out here, are going to be largely hydrophobic. But recognize, too, that because it's a spiral staircase, that as you would walk around the spiral staircase, some of these amino acids will have their R groups pointing inward. Predominantly, now it's not always the case, but predominantly we find these R groups that are pointing inward are going to be polar or charged. If these R groups pointing inward are polar or charged, they now can interact with water. Because they can interact with water, they provide a hydrophilic pathway across the phospholipid bilayer. So now we have a mechanism whereby we can organize these alpha helical regions. We can choose the R groups we put on those alpha helical regions and make a particular kind of transmembrane channel that allows sodium or potassium or calcium to selectively move from the outside of the cell to the in or the inside to the out. The characteristic of these transmembrane proteins that are channels is that they contain hydrophilic amino acids pointing inward. Into the channel. So now we have walked around the problem of having this barrier, this hydrophobic barrier. And we've now made a selective pathway that extends across the lipid bilayer. What's more is out here in the extracellular region and the intercellular region, we have regions of that protein which are living in either the cytoplasm or the extracellular environment, both of which are hydrophilic. That means by selecting the appropriate R groups and organizing the alpha helical and beta pleated sheets in just the right way, we can get a specific three-dimensional shape on the inside of the cell and on the outside of the cell. This becomes particularly important when we're thinking about transmembrane proteins that are functioning as channels that can be gated, can be opened or closed. Because you can imagine if we have a particular three-dimensional shape out here, let's say right in this site, we could then at this site bind something called acetylcholine, or a neurotransmitter called glutamate, or any number of other molecules that can now bind to this region on the outside of the cell, and then we'll find later on in the course operate gates in the middle of the channel to open or close them, and then regulate the movement of ions in and out of the cell. We'll spend quite a bit of time on that when we talk about the nervous system, but for now simply recognize that what we have are transmembrane regions that are predominantly alpha helical, and the combination of hydrophobic R groups pointing toward the hydrophobic tails of the phospholipids and hydrophilic R groups pointing inward toward one another, we can now make a transmembrane region that generates a proper channel, a proper hydrophilic channel, and that by having a particular amino acid sequence with its three-dimensional structure in the outside and the inside of the cell, we can further provide means of operating these channels and regulating these channels by binding of molecules, extracellular and intracellular, through the three-dimensional shape. Any questions to there? Yes?
Can you make a transmembrane channel with only one alpha helix, or do you need 2+?
So the question here is, can you make a transmembrane channel with one alpha helix? As far as I know, the rule of thumb is you need at least 4. But 4 isn't all that common either. What we'll find is that when we talk about voltage-gated channels, we get to the nervous system, those have 24 transmembrane regions. And there are 4 sets of 6. When we talk about something like adenylyl cyclase, which is embedded in the membrane, I think it has 12 or more transmembrane regions. When we talk about the ligand-gated channels, like the acetylcholine receptor, the acetylcholine receptor has 5 subunits, each of which crosses the membrane 4 times. So you get into these really complex organizations of how you thread the alpha helices across the membrane, but in all cases the channels are generated from alpha helix. Right. But you're right, it's not just 1, it's not just 2. 4 is a minimum, and then it gets to a bigger number after that. Yes?
Beta barrels across, or is that just not relevant?
So the beta barrel question. This is one that comes up typically at this point. Yes, you can do this with beta barrels. Turns out the only creatures that do beta barrels are prokaryotes, and that they tend to do this in their outer membranes when they're double membranes. You find some beta barrels apparently in chloroplasts and in mitochondria as well. But again, we'll see here a little later that both chloroplasts and mitochondria have their ancient origins as prokaryotes. So the beta barrel form of a channel seems to be relegated to prokaryotic organisms, whereas by the time we begin to see eukaryotic organisms, we use alpha helices. If we play the tapes all over again, maybe eukaryotes could have beta sheets too, but so far we don't.